Think AB blood type is simply what happens when A and B meet. That is the textbook version. It is also only the beginning. Because if you carry AB blood, your red blood cells are displaying something unusual. Two different molecular signatures at the same time. And those signatures are not random decorations. They are the visible result of ancient variation inside a single genetic system that has been changing, recombining, and surviving across evolutionary time. Your blood type is written into your DNA. And AB is the phenotype that appears when you inherit a functional A allele from one parent and a functional B allele from the other. Simple. Except the history behind those two alleles is anything but simple. The ABO gene contains three major allele forms, A, B, and O, and the A and B versions produce enzymes that modify the same underlying precursor on the surface of red blood cells in different ways. The O allele generally produces an inactive enzyme, leaving that precursor unchanged. So when you are AB, your cells are not choosing between A and B. They express both. That single fact opens a much larger story involving ancient primates, human migrations, infectious disease, population genetics, and one of the oldest genetic mysteries in medicine. And the strangest part is this. The A and B variants are not merely recent human inventions. Evidence suggests that the evolutionary history of the ABO polymorphism reaches far beyond our species. Researchers have found A and B forms in multiple primate species, with the A-B polymorphism estimated to have persisted for roughly 20 million years or more. 20 million years. Long before modern humans. Long before cities. Long before agriculture. Long before anyone had a concept of blood type. That's not a coincidence. It's a signal. The answer touches molecular genetics, ancient evolution, disease, migration, and the hidden reason nature may have preserved multiple blood group variants instead of allowing one to take over completely. And it starts with a discovery made more than a century ago that transformed medicine forever. Stay here, because by the end, AB will look less like a simple blood label and more like a surviving fragment of an ancient evolutionary experiment. For most of modern history, nobody knew that blood came in different genetic types. Blood looked like blood. Then, around 1900, an Austrian physician named Karl Landsteiner began mixing blood cells and serum from different people. Some combinations reacted. Others did not. The reactions were caused by molecular structures on red blood cells and antibodies in plasma. Landsteiner's work eventually led to the ABO blood group system, one of the foundations of modern transfusion medicine. But the deeper genetic explanation came later. By 1910, researchers had demonstrated that the A and B characteristics were inherited, and that they behaved as codominant traits relative to O. In 1924, Felix Bernstein's three-allele model provided the framework that explained how three major alleles could produce four basic blood group phenotypes. A, B, O. Three major alleles. Four common phenotypes. A, B, AB, and O. And AB is where the inheritance pattern becomes especially interesting. You carry two ABO alleles. One comes from your mother. One comes from your father. If you inherit A and O, your phenotype is generally A. If you inherit B and O, your phenotype is generally B. If you inherit O and O, your phenotype is O. But if you inherit A and B, neither one simply disappears. Both are expressed. You become AB. That is codominance. And it means your blood cells can display both A and B antigens. Sit with that for a second. Your phenotype is not telling you that your parents had the same blood type. It is telling you that two different inherited instructions survived together in your genome and both remained functional. But that raises a deeper question. Where did those instructions come from in the first place? Because the modern ABO gene is not just three neat options sitting inside human DNA. It is extraordinarily diverse. The ABO locus is located on chromosome 9. Its major alleles produce different versions of an enzyme called a glycosyltransferase. That sounds technical, but the basic idea is surprisingly simple. Imagine the surface of a red blood cell as a landscape covered with molecular structures. The ABO enzyme acts like a tiny molecular construction worker. The A version attaches one particular sugar. The B version attaches another. The O version, in its common form, has a mutation that disrupts the enzyme's function, so it cannot perform the same modification effectively. A and B therefore represent different biochemical outcomes, and the difference between them can be traced to changes in the DNA sequence. Researchers have identified 4 key amino acid substitutions that distinguish the A and B transferases in the classic forms of the gene. 4 amino acid differences. That is all it takes to alter the molecular specificity of the enzyme. Not an entirely different gene. Not an entirely different chromosome. A handful of molecular changes inside one genetic system. But the story becomes even stranger when scientists stop looking at the phenotype and start comparing the DNA itself. Because A and B are not simply modern human versions of 2 unrelated inventions. Their evolutionary history appears to be much older. Travel backward. Not thousands of years. Not hundreds of thousands. Millions. The ABO blood group system is not unique to humans. A and B variants have been identified in at least 17 other primate species, and researchers studying the evolutionary history of the gene have argued that the A-B polymorphism may have been maintained for around 20 million years or more. That means the biological machinery behind your AB blood type has an evolutionary history that predates Homo sapiens by an enormous margin. Your blood is carrying a system that existed before humans were humans. Let that settle. This changes the question. The question is no longer simply why some people are A, some are B, some are O, and some are AB. The deeper question is why evolution kept multiple versions around for so long. Usually, when a genetic variant becomes strongly disadvantageous, natural selection can push it downward. When another variant provides a consistent advantage, it can become more common. But ABO does not fit neatly into that simple picture. Instead, A, B, and O have remained in human populations at different frequencies. That pattern has led researchers to investigate balancing selection and frequency-dependent selection as possible explanations. One influential review notes that the ABO gene has unusually high genetic diversity and suggests that fluctuating selection pressures, potentially involving interactions with pathogens in the gut, may help explain why different variants have persisted. But there is an important qualification. Scientists have not identified one universal evolutionary explanation that explains every ABO distribution everywhere. The exact selective forces remain debated. That uncertainty matters, because science is not strongest when it pretends every mystery has already been solved. Science is strongest when the evidence tells us what we know, and where the boundary of knowledge begins. And the distribution of AB blood gives us another clue. AB is uncommon compared with A, B, and O. The exact frequency varies dramatically between populations. Older NCBI reference data, for example, report AB frequencies of roughly 3 to 5% in several broad population categories, with differences between A1B and A2B subgroups. More recent population studies show that frequencies can differ considerably depending on the population sampled. In a multi-center study of 10,000 healthy blood donors in India, AB accounted for 7.74% of the sample, while O accounted for 37.12%, B for 32.26%, and A for 22.88%. And in a recent study comparing university students in Liberia and Pakistan, AB occurred in 7.4% of the Liberian sample and 8% of the Pakistani sample. Those numbers are not contradictory. They demonstrate something important. Blood group frequencies are population-specific. Geography matters. Ancestry matters. Migration matters. And population history matters. Take Pakistan. The recent Liberia-Pakistan comparison found B as the most common group in its Pakistani sample, followed closely by O and A, with AB at 8%. Look at another Pakistani dataset. A study involving 30,682 people from Saftrabad and Faisalabad reported AB as the least frequent of the 4 major ABO groups in that dataset, although its observed prevalence was 14.83% in the Faisalabad group reported by the study. Different populations. Different sampling frames. Different numbers. The lesson is not that 1% is the universal AB number. The lesson is that your blood type exists inside a population history. And that population history is written partly in the frequencies of alleles passed from one generation to the next. But there is another layer. How did the A and B alleles themselves evolve? For years, researchers assumed a relatively straightforward evolutionary relationship among the major ABO alleles. The A and B forms differ in critical positions within the gene. The major O allele has a characteristic deletion that disrupts the resulting enzyme. But molecular studies revealed a much more complicated picture. A 2013 analysis using a larger HapMap SNP dataset examined a hypothesis that the modern human A lineage may have emerged through recombination involving B and O-related sequences. The authors argued that specific haplotypes supported a recombinant history, while also noting that other studies proposed different evolutionary models, including ancient maintenance of A and B polymorphisms across species. That distinction is important. The evolutionary history of ABO is not a clean family tree where every branch has been permanently settled. It is closer to a tangled genetic history. Mutation, recombination, selection, drift, migration, population separation, then contact again. The gene preserves traces of all of these processes. And AB is the phenotype that lets you see two functional sides of that history at once. One inherited A. One inherited B. Two molecular instructions. One blood type. But the story does not stop with inheritance. Because scientists eventually discovered that ABO variation interacts with something far more powerful than family trees. Disease. Your red blood cells are not isolated from the rest of your biology. ABO antigens can also be expressed on other cell types and tissues, and ABO-related molecular structures interact with biological systems involved in infection, inflammation, and vascular processes. Researchers have therefore investigated associations between ABO groups and numerous diseases. But association is not the same as destiny. That distinction matters. A particular blood group can be statistically associated with a higher or lower risk for a disease without meaning that the blood group directly causes the disease. The evolutionary question is even harder. If pathogens helped shape ABO diversity, which pathogens? When? In which populations? And under what environmental conditions? There is no single answer. Researchers have proposed different mechanisms involving host-pathogen interactions, including pathogens that interact with carbohydrate structures related to ABO antigens. But this remains an area where evidence is complex. One population may experience a different infectious environment from another. A variant that is advantageous under one set of conditions may be neutral or disadvantageous somewhere else. That creates a moving target for natural selection. And that may be one reason multiple ABO variants survived. The environment changes. The pathogens change. Populations move. Selection changes. The genetic system responds across generations. Not perfectly, not predictably, but persistently. That is the deeper story behind your blood type. And there is one more piece that makes AB especially interesting. The same system that helps scientists reconstruct ancient evolution is still being refined by modern genomic technology. Before we get there, if you care about the hidden genetic stories inside ordinary human traits, subscribing keeps these investigations going. Because the more closely scientists examine the genome, the less simple our ancestry becomes. Now consider what happened when researchers stopped studying only the classic A, B, and O categories. They found many more versions hiding underneath. The ABO gene contains enormous genetic diversity. A historical review by Fumihiko Yamamoto describes how molecular work in 1990 helped establish the genetic basis of the ABO polymorphism, showing the relationship between the A, B, and O alleles and the enzymes responsible for producing their corresponding antigen structures. Later research identified many additional subgroups and variants. A1, A2, A3, X, B3, cis-AB, BA, and many others. The old four-letter system is therefore a useful shorthand. But it is not the entire genetic story. Modern sequencing has pushed that point even further. Researchers have identified hundreds of ABO alleles and numerous sequence variations across the gene. That means two people who are both classified as AB can still carry different molecular versions of the A and B alleles. Same phenotype. Different underlying genotype. Same label. Different DNA. That is a recurring lesson in genetics. The visible trait is often only the surface. The genome underneath is more complicated. And sometimes that complexity changes the medical picture. There is another genetic system sitting beside ABO that can completely change how we interpret a blood type. The H antigen. A and B antigens are built from a precursor structure known as H antigen. Without the appropriate H structure, the A and B modifications cannot be produced normally. In rare individuals with the Bombay phenotype, mutations affecting the FUT1 pathway prevent the normal expression of H antigen on red blood cells. As a result, the cells also lack the ordinary A and B antigens, even when the person's ABO genotype would otherwise suggest A, B, or AB. This is where the simple blood type chart begins to break apart. Someone can genetically carry information associated with A or B and still present an unexpected blood phenotype because another part of the biochemical pathway is disrupted. The label is not the whole biology. The pathway matters. The molecular mechanism matters. The genome matters. And that is precisely why DNA changed the study of blood groups. It moved scientists from asking, "What does this person's blood look like?" to asking, "What sequence produced this phenotype, and what else is happening around it?" That shift is enormous, because once you can read the sequence, you can begin reconstructing history. Imagine two populations separated for thousands of years. Their environments differ. Their diseases differ. Their migrations differ. Their population sizes differ. Over generations, allele frequencies change. Some variants become more common. Others become rare. Some disappear locally. Others arrive through migration. Then populations meet again. The modern frequency of A, B, O, and AB can therefore contain traces of population history. Not a perfect record. Not an ancestry certificate. But a statistical signal. That distinction is crucial. You cannot look at someone's AB blood type and identify a precise ancient tribe. You cannot infer someone's complete ancestry from ABO alone. And you certainly cannot use blood type to establish racial superiority or infer a hierarchy of human populations. Human genetic variation does not work that way. The ABO system is one locus among thousands. Your ancestry is vastly more complicated. But one locus can still tell a story. And ABO has been telling its story for an exceptionally long time. Return to the molecular level. Your AB blood cells carry A and B antigens. The A and B alleles encode different glycosyltransferase activities. The two alleles are expressed together. The result is the AB phenotype. That sounds almost mechanical. But behind it is an evolutionary history stretching across primates. Behind that history are mutations and recombination. Behind those mutations are population movements. Behind those population movements are changing environments. And behind some of the evolutionary hypotheses are pathogens that may have exerted selective pressure on our ancestors. Layer after layer. The blood type is the final visible result. But the process that produced it is ancient. Very ancient. And there is something almost strange about that. Your body is manufacturing these molecular structures right now without you thinking about them. Every heartbeat. Every circulation of blood. Every ordinary day. A genetic system that predates our species is quietly operating inside you. That is not metaphor. That is biology. But there is still something scientists do not know. They do not have one universally accepted explanation for exactly why the A, B, and O polymorphism has remained so persistent across such deep evolutionary time. Balancing selection is a compelling framework. Host-pathogen interactions are plausible contributors. Frequency-dependent selection may help explain why the advantage of one variant could depend on how common other variants are. But the complete history is more complicated than a single sentence. Researchers continue to debate the relative importance of selection, demographic history, mutation, recombination, and other evolutionary forces. That uncertainty is not a failure. It is the interesting part. Because every new genome gives scientists another piece of the puzzle. Modern population-scale sequencing has already revealed detailed differences in ABO allele frequencies across large populations. One study analyzing more than 113,000 German samples, for example, estimated allele group frequencies of roughly 29.5% for A, 9.6% for B, and 61% for O in that dataset. Numbers like these let researchers move beyond 4 labels. They can examine specific alleles, specific mutations, specific haplotypes, specific population histories. The blood bank sees AB. Genomics sees much more. And this brings us back to you. If your blood type is AB, your body is expressing both sides of the classic A-B system. That does not make you biologically superior. It does not reveal a secret race. It does not identify a lost civilization. It does not provide a complete ancestry report. What it does reveal is something more scientifically meaningful. Your genome is carrying a combination of inherited variants that have survived through countless generations of reproduction, migration, population change, and environmental pressure. You inherited one side from one parent, another from the other, and both remained visible. 2 ancient molecular pathways, 1 phenotype, 1 body, 1 living descendant of an evolutionary history far older than human civilization. Think about what that means. The blood moving through your veins is not simply carrying oxygen. It carries information. Not a readable autobiography. Not a perfect map. But fragments of biological history. Your ancestors did not know what ABO was. They did not know chromosomes existed. They did not know what an allele was. They simply survived, reproduced, migrated, mixed, separated, and passed their DNA forward, generation after generation, until one generation eventually produced you. And inside your cells, that ancient genetic history is still operating. The label says AB. The DNA tells a longer story. A story that begins before you, before your parents, before your grandparents, before recorded history, before Homo sapiens had written language, and perhaps before the branch leading to modern humans had even taken its present form. That is the real truth about AB blood. It is not that you have 2 blood types. You don't. You have 1 phenotype produced by 2 codominant alleles. But those alleles belong to a genetic system whose history is far older, more complex, and more interconnected than the 4 letters on a medical chart suggest. Common labels simplify biology. DNA restores the complexity. And every time scientists read another genome, another piece of that ancient history becomes visible. You are not carrying a simple blood type label. You are carrying 1 surviving chapter of a genetic story millions of years in the making. The blood remembered the history. DNA finally gave us a way to read it.